Welcome to this week's session of anatomy. So we're going to start looking at the organization of the nervous system. So this week we're going to look at the organization of the nervous system. Now, the nervous system, as you may already know, is that system that is responsible for transmission of impulses, electrical impulses, from one part of the body to another. Okay, it is that system that is responsible for transmission of impulses from one part of the body to another. Okay, the main cells of the nervous system, the cells that carry out this function of transmitting impulses from one part of the body to another, are known as the neurons. The neurons are the parenchyma of the nervous system. Okay, so the nervous system is made up of two types of cells, okay, structurally. And these cells include one, the neurons themselves. Two, we have what are known as glial cells. Glial cells are supportive cells of the nervous system. So these glial cells, we have several different types of glial cells. We have 6 types of glial cells. Two of them are found in the peripheral nervous system, and 4 of them are found in the central nervous system. Okay, so we are saying that neurons are grouped as circuits in the nervous system, meaning they connect from one neuron to another. Okay, so that is how impulses even move from one neuron to another, from one organ to another, just like that. Now, let us look at the glial cells. We are saying that we have glial cells that are responsible for the um, for the support of the nerve tissue, support of the neurons. Okay. Now these glial cells, we said that we have about 6 glial cells. One, we have oligodendrocytes. Oligodendrocytes are normally found in the central nervous system. They are normally found in the central nervous system where there is um, where the brain and spinal cord are located. That is where you're going to find oligodendrocytes. Their origin is the neural tube. Remember when we did embryology, we talked about the neural tube. Okay, now that neural tube is where the oligodendrocytes will originate from. And then we have their function is to produce myelin. So their function is to myelinate the neurons of the central nervous system. That is the function of the oligodendrocytes. And then we have a Schwann cell, which performs the same function as the oligodendrocyte. The difference is that it is found in the peripheral nervous system. Instead of being found in the central nervous system, it is found in the peripheral nervous system, and its function is to also produce myelin for these cells. So it's to myelinate the neurons. That is the function of a Schwann cell. And then we have astrocytes. Okay, we have astrocytes. They are also found in the nervous, in the central nervous system, and their origin is also from the neural tube. Their origin is also from the neural tube. Their function is to give structural support to the neurons, also to repair any worn out tissues or neurons if the damage is not too much, and they are the ones that are responsible for formation of the blood-brain barrier to regulate what goes inside the neurons from the blood and what should not be allowed to go into the neurons from the blood. Okay, so those are the astrocytes. And then we have ependymal cells. The ependymal cells also originate from the neurons. And these ependymal cells are found in the central in the central nervous system. They normally line the cavities, okay, those cavities of the central nervous system. So, for example, the lateral ventricles, the third ventricle, the fourth ventricle, they are the ones that line those ventricles. Now, among the ependymal cells, we are going to find some of these ependymal cells that are uh, normally specialized for secretion of cerebrospinal fluid, for the secretion of cerebrospinal fluid, and those cells form what are known as the telachoroidea. Okay, they form what is known as telachoroidea. And then we have microglia. Microglia are said to be the uh, they are said to be the white blood cells, the macrophages of the nervous system. Remember, we said that when we were looking at tissues, we said that white blood cells, specifically the macrophages, these macrophages they adopt a different name depending on which organ they are in. So, for example, we did say that the macrophages of the liver will be called the Kupffer cells. Okay, therefore, even in the central nervous system or in the nervous system, we are going to find that the macrophages will have a different name. And so that name for these macrophages in the nervous system is microglia. So we are saying that the name for the macrophages in the central nervous system are known as the microglia. The sixth cell that has not been mentioned here is is a satellite cell. Satellite cells also support the nerve tissue. They are found in the peripheral nervous system and usually found along the bodies of the sensory neurons. They also support um, they also support nerve tissue. So those are the supportive cells of the nervous system. Now, the divisions of the nervous system. We have two major divisions of the nervous system. Two major divisions of the nervous system. One, we have the central nervous system, and the central nervous system is said to be made up of the brain and the spinal cord. Okay, the central nervous system is said to be made up of the brain and the spinal cord. The peripheral nervous system, on the other hand, is made up of all the other neural tissue that you're going to find outside the central nervous system. So this would include all the cranial nerves, so all the 12 cranial nerves, because remember, even though they originate from the brain, they still work from the peripheral nervous system and the spinal nerves as well. So all the nerves and all the nerve tissue that you're going to find outside the brain and spinal cord, that is what we're going to call the that is what we're going to find in the peripheral nervous system. So those are the two major divisions of the nervous system. The other thing that you need to take note of is what we call cell bodies. Okay, so cell bodies of neurons form gray matter in the central nervous system. So when you hear someone say the gray matter, then we're talking about the cell bodies of neurons that are found in the central nervous system. Now, when you look at these groups of cell bodies, they have a different name depending on where they are. So the cell bodies that are found inside the central nervous system, so either in the spinal cord or in the brain, then we are going to refer to them as nuclei. So one cell or one group of cell bodies is going to be called a nucleus. Several groups of cell bodies uh, is going to be known as it's not going to be known as are going to be known as nuclei in the central nervous system. However, in the peripheral nervous system, cell bodies that are in groups are known as ganglia. So we're going to find several different types of groupings of cell bodies, and those will be known as the ganglia. Okay. Now, physiologically, that is in terms of function, physiologically, this the peripheral nervous system can then be divided into efferent division and an afferent division. Okay, an efferent division and an afferent division. So we are saying that the efferent division is the one that carries signals away from the central nervous system. The efferent division is the one that carries impulses away from the central nervous system. So when you hear the word efferent, we are simply talking about carrying away from, away from. So efferent means those neurons that carry impulses away from the central nervous system. The other way that you can refer to these neurons would be that they are motor neurons. These are the ones that take information to the effector organs. The effector organs, which are going to do something once they receive information. For example, the muscles, they're the ones that are going to contract so that there is movement. Okay? And then we have the afferent division. The afferent division, these are neurons which bring information from the periphery to the central nervous system. These, therefore, should be the sensory neurons. These neurons are the ones that bring sensation, whatever it is, whatever it is that you have felt as a person, whether it is heat, you have touched something hot, or someone is touching you, or you have pricked yourself, whatever that information is, it's going to be carried to the brain through the afferent division of the nervous system. And those cells, those neurons that make up the afferent division, are then said to be sensory neurons. They are then said to be sensory neurons because they are the ones that are sensing, okay? They are the ones that are sensing these impulses. They are the ones that are sensing these impulses. So when you look at the nervous system topographically, when you look at the nervous system at a glance, you will notice that when the nervous system is made up of the central nervous system and the peripheral nervous system, and the central nervous system, like we've already said, is made up of brain and spinal cord. The peripheral nervous system is made up of 12 pairs of cranial nerves and 31 pairs of spinal nerves. Okay? And then there is another system that some people, some books will refer to as part of the peripheral nervous system, but other books will specify and say it is a nervous system on its own. And so this is known as the enteric nervous system, which is the nervous system of the gastrointestinal tract. Okay, the nervous system of the gastrointestinal tract. So these are the divisions of the nervous system. When you look at this, we are talking about having a whole nervous system, which is divided first of all into peripheral nervous system and central nervous system. Then the peripheral nervous system is further divided into efferent division and afferent division, like we have already discussed. Now, the efferent division is that division that takes away impulses from the central nervous system to the effector organs. Okay, it is further made up, it is further divided into an autonomic system, which is an autonomic division, and a somatic division. The autonomic division is usually supposed to innervate organs that are autonomic in nature. Those organs that are going to just function without your concern. For example, the heart, okay? For example, the smooth muscles, for example, the glands. So that is where it's going to innervate. And then we have the somatic system. The somatic system is the one that comes to your voluntary muscles, the muscles of your hands, the muscles of your legs, those muscles that you can tell to say, I want to stand up, and then they agree and you stand up, okay? As opposed to the autonomic system that innervates organs that are involuntary. For example, the intestines, whether they like it or not, they have to start churning the food. They have to start digesting the food, whether they like it or not, as long as they've been told to do so. Okay? Now, this same autonomic nervous system is further subdivided into the sympathetic nervous system and parasympathetic nervous system. The sympathetic nervous system, I'm sure we already know, is also known as the flight and fight nervous system. This one activates all the involuntary functions that will make you ready to fight or to run. So, for example, it is going to divert all the energy from digesting the food that you have eaten, but it is going to bring the energy to the muscles of, let's say, the legs, so that you start running. It is going to activate the muscles in the iris so that your pupil will dilate and in an effort to allow you to see more clearly as you run away from whatever danger, okay, you are running from. So that is what the sympathetic nervous system is. The parasympathetic nervous system, on the other hand, is known as the rest and digest system. This is the system that functions when you are at rest and allows for your body to still go ahead and work even as you are at rest. Okay, so that parasympathetic nervous system is also known as the rest and digest, nervous system. Okay, now when you look at the the way the spinal cord is divided, we have certain nerves that come from. Remember when we did the spinal cord, no, not the spinal cord, when we did the vertebral column, we did mention that the vertebral column is made up of several vertebrae, and we say that we have 7 vertebrae in the neck. Those are cervical vertebrae. We have 12 thoracic vertebrae. We have 5 lumbar vertebrae. We have 5 sacral vertebrae and 4 coccygeal vertebrae. So at each point between these vertebrae, we are going to have nerves emerging from the spinal cord, coming into the peripheries. Okay, so we're going to have 31 pairs of spinal nerves. That means on the right one, on the left one. So 31 pairs of spinal nerves. And then on the head side, those nerves that are responsible for the head and neck, those are the cranial nerves. So those we are going to have 12 pairs of cranial nerves as well. Okay. Now, among those 31 pairs and 12 pairs of these nerves in the peripheral nervous system, we are going to find that some nerves are the ones that carry nerves that have sympathetic stimulation and others do not. So others will carry parasympathetic nervous stimulation, and then we'll have others that carry sympathetic nervous stimulation. So the ones that carry sympathetic nervous stimulation are the thoracic nerves, okay? Thoracic nerves. So now the thoracic nerves plus the first two lumbar nerves, okay? So lumbar, you remember there were five vertebral bones, so five lumbar bones, but the top two nerves coming from there. Now, even the nerves, by the way, are named according to the name or the names of the bones. So, for example, in the thoracic region, we're going to have T1, T2, T3, T4, okay, until we go to T12. Um, those are the thoracic, those are the thoracic vertebrae, and so you're going to find that even the neurons will be uh, will be following that kind of a pattern. And there we are going to call them intercostal nerves, okay? So the outflow from the thoracic vertebra plus two the top two lumbar vertebra this is where the sympathetic nerves are. This is where sympathetic stimulation, where you're going to find nerves that have sympathetic stimulation in them. Okay, this is where you're going to find nerves that have sympathetic stimulation in them. And then we have the parasympathetic stimulation. Those ones come from what is known as the craniosacral outflow. The craniosacral outflow includes some of the cranial nerves, and these cranial nerves that that help to make up the parasympathetic system. The craniosacral outflow include cranial nerves 3, okay, cranial nerves 7, cranial nerve 9, and cranial nerve 10. So those are the ones that are going to help. So we have oculomotor, cranial nerve 3, facial, cranial nerve, um, you know, 7. So these are the ones that will help, as well as S4 to S2 to S4. So we have sacral 2 to sacral 4. So sacral 2 to sacral 4 and cranial 3, 7, 9, and 10. These are where we have the craniosacral outflow, and that is what is going to make up the that is what is going to make up the parasympathetic nervous system. Okay, so now let's look at the central nervous system in detail. We said that the central nervous system is made up of the brain and the spinal cord. So we'll start looking at the brain, then we'll look at the spinal cord. Let us look at the gross structure of the brain. So when you look at the brain, this picture is a superior view. You're looking at this human being on top of their head, okay? On top of their head. You will notice that the brain on top of the head is going to look like it is divided into two. There is this side on the left and this side on the right. And so you're going to say it is made up of two hemispheres. Now, these two hemispheres, they only apply to the cerebrum of the brain. We're going to look at all that. So there are these two cerebral hemispheres, the left and the right. They are divided in the middle by this very deep fissure. Okay, very deep fissure, and this is known as the longitudinal fissure. The other thing that you will notice is that the cerebrum is going to be in a lot of folds. So all these many folds, okay? These folds, all these folds. Now these folds are known as gyri. One is a gyrus, okay? And then a number of them are gyri, okay? One is a gyrus, and all these gyri, they all have names. Each gyrus has a name, okay? But just take note that they are known as the gyri, okay? And then those small furrows between the gyri, okay? Those are known as sulci. So one is a sulcus and several of them are sulci. Okay. So now we're saying the brain is contained by the neurocranium, okay, composed of the cerebrum, the cerebellum, and the brainstem. So if you look at this, this is one hemisphere, this of the cerebrum, okay, the cerebellum, and we have the brainstem here, okay. So the brain has gyri, as I was telling you, and sulci, all right? And then we have the cerebrum, uh, include the cerebral hemispheres and the basal ganglia. So we are saying that the cerebrum, which is going to include the cerebral hemispheres, this is one hemisphere. So we're looking at this picture after having dissected the brain in the mid-sagittal region. And so you're looking at one side of the brain. So this one hemisphere is part of the cerebrum, okay? It's part of the cerebrum. And then we are going to find that we have several bodies of neurons, several nuclei of neurons there. And those nuclei will be, when you collectively, they are known as the basal ganglia or basal nuclei. The cerebral hemispheres are separated by the four cerebri. They are separated by the four cerebri. There is some kind of epithelial tissue, a tent-like tissue that comes to separate these two, which is found in the longitudinal fissure. Okay, that one is from a meninge known as the dura mater, and then it comes and just comes and is between the two hemispheres. Each cerebral hemisphere is divided into four lobes. Each cerebral hemisphere is divided into four lobes, each of which is related to the overlying bones of the same name. Remember when we did the bones of the skull? So their names are also going to relate even to the lobes of the brain. So from the superior view, the cerebrum is essentially divided in two quarters by the median longitudinal fissure and the central sulcus. So we have this longitudinal fissure over here, which divides the brain into half. However, in approximately the mid-coronal area here, you're going to have the central sulcus, okay? Therefore, we are going to say that the whole brain here is divided into four, and those are quarters. There is this quarter here, this quarter here, this quarter here, and this quarter here. So the central sulcus separates the frontal lobes posteriorly, anteriorly, and the parietal lobes posteriorly. Remember, if we're going to name these lobes according to the bones, remember we had the frontal bone, which makes up our forehead. Then we said we have just posterior to the frontal bone, we have parietal bones, and then we have the occipital bone over here. So now, in the lateral view, this was the superior view, and then we have the lateral view over here. In the lateral view, these lobes lie superior to the transverse lateral sulcus. So these two lobes will lie superior to the transverse lateral sulcus. The lateral sulcus is the one that separates the parietal lobe and the frontal lobe from the temporal lobe here. So the lateral sulcus here is what will separate the parietal and the frontal lobes from the temporal lobes. Then posteriorly, the posteriorly placed occipital lobes are separated from the parietal and temporal lobes by the plane of the parietal occipital sulcus over here. Okay, the parietal occipital sulcus. The anteriormost points of the anteriorly projecting frontal and temporal lobes are frontal and temporal poles. Okay, so if you look at this picture, you are going to notice that this is the posterior aspect, this is the anterior aspect. The frontmost part, that is the frontal pole, and the frontmost part of the temporal lobe is the temporal pole. The posteriormost part is what is going to form the occipital pole. So the posteriormost. So if you're looking at this brain, we are saying this is the anterior aspect. This is the posterior aspect where you're going to find the cerebellum. The posteriormost, part is what is going to be known as the occipital pole. The diencephalon forms the central core of the brain. So we're going to notice that here we are going to see what is known as the diencephalon. Okay. Uh, the pons is the part of the brain of the brainstem between the midbrain rostrally and the medulla oblongata caudally. So we are saying that this is what we are going to call now the brainstem. Okay, so we have the cerebrum, we have the diencephalon here. The diencephalon has several other things, the thalamus, the epithalamus, the hypothalamus, okay? And then we have the brainstem over here. The brainstem. And the brainstem is composed of the midbrain, the pons, and the medulla oblongata. The medulla oblongata is the one that is continuous with the spinal cord inferiorly. So the medulla oblongata is the most caudal, meaning it's the most inferior subdivision of the brain that is continuous with the spinal cord. The cerebellum is the large brain mass lying posterior to the pons and medulla. Okay, so the pericerebellum is here. It lies posterior to the pons and the medulla and inferior to the posterior part of the cerebrum. So it is inferior to the posterior part of the cerebrum. It is posterior to the pons and the medulla. So that is what we are saying. All right. Now, since we've looked at the parts generally, okay, of the brain, let us quickly look at the ventricular system of the brain. We will notice that the brain has cavities. The brain has several cavities through which a fluid known as the cerebrospinal fluid flows, and where this cerebrospinal fluid is produced. So we have four cavities. Four cavities. First of all, we have lateral cavities. These are two of them, one on each side of the cerebral hemisphere. And then we have one midline a third ventricle over here. In this third ventricle, on either side of the ventricle, you're going to find that is where the thalami are. One thalamus on either side. So on the right, we have the thalamus. On the left, we have the thalamus. In the middle, okay? In the middle, that is where we're going to find the third ventricle. It is connected, the cerebrospinal fluid flows from the lateral ventricles to the third ventricle through the interventricular foramen over here, okay? The other ventricle that you will find will be the fourth ventricle. The fourth ventricle is located posterior to the pons, okay, to the pons and so posterior to the lower part of the pons and the upper part of the medulla, and then anterior to the cerebellum. Okay, so these are the four cavities. The connection between the third um, and the fourth ventricle is the cerebral aqueduct. Cerebral aqueduct. That is the connection between the third and the fourth ventricle. Okay. Now, secretion of CSF, that is cerebrospinal fluid. So cerebrospinal fluid is secreted at the rate of 400 to 500 mils a day by the choroidal epithelial cells. These are modified ependymal cells, like we already talked about. Now, this happens in the lateral third and the fourth ventricles. All right, in the lateral, in the third, and in the fourth ventricle. So this diagram here is just simply showing you how CSF is flowing, okay, all the way from the lateral ventricles, all the way to the third ventricle, all the way to the fourth ventricle, okay? That is how cerebrospinal fluid flows. So we are saying that the choroid plexuses are fringes of vascular pyramids. Okay, we are going to look at these meninges very soon. The vascular pyramater, which is said to be the telachoroidea. Now, this telachoroidea is covered by cuboidal epithelial cells into the roofs of the third and fourth ventricles and on the floors of the bodies and inferior horns of the lateral ventricles. So you're going to find this, okay, the choroid plexus, all right? That is where cerebrospinal fluid is produced. All right. Let's now look at the cranial meninges. Meninges are membranous coverings of the brain, and these lie immediately internal to the cranium. So the cranium being the skull. Okay, so immediately internal to the cranium, we're going to find these membranous coverings known as the meninges. These meninges protect the brain, okay? They form supporting framework for arteries, blood vessels. They pass through either the meninges themselves or the spaces between the meninges. The other function is that they enclose a fluid-filled cavity. This cavity is known as the subarachnoid space. So that cavity is said to be the subarachnoid space. The subarachnoid space is vital to the normal function of the brain. Okay, very, very, very vital. So when you look at these meninges now, we have three meninges. So uh, if you look at if we look at this picture here, we are going to notice that this is the this is this piece was taken from the head of someone. Okay, just a tiny piece. So this is the bone making the skull, all right? Making the skull, okay? And then we have the dura mater. The dura mater is the layer or one of the meninges which lines, this is the outermost layer, okay? This is the outermost layer of the uh, of the three layers, is the outermost layer. Now we are saying that this dura mater is further divided into two layers. There is this one layer which is adherent to the skull, which is adherent to the skull bones, which is adherent to the cranial bones. This layer is the periosteal layer. However, the dura mater, there are some points where it meets itself, but at most points, it kind of separates, okay? It kind of separates, leaving a space inside, okay, between itself. So this other layer of the dura mater that goes into the brain on the inner aspect is what is going to be called the meningeal layer. Okay, the one that flows together with the arachnoid matter. This is the meningeal layer. So now you may want to find out what fills the layer between the two layers of the dura mater. We have venous blood. We have venous blood that is draining from the brain, okay, with less oxygen, with less nutrients. This is the venous blood. So we have the meningeal layer over here, and we have the dura layer over here, and in the middle we have the uh, space filled with the venous blood. Now, this, if you've noticed here, it's saying superior sagittal sinus. This one was taken from the front of the person. So this furrow here is going to be the longitudinal sulcus that we talked about. So this is going to be one cerebral hemisphere, and this is going to be another cerebral hemisphere. So this meningeal layer, the meningeal layer of the dura mater is the one that is going deep into the cerebral, uh, into the longitudinal sulcus. And that folding inside is what is known as the falx cerebri or fox cerebri. Okay, that is what is going to be known as the falx cerebri. Now, just internal to the dura mater, you are going to find another layer, which is known as the arachnoid mater, which is known as the arachnoid mater. Okay, it is just the thin intermediate layer, the thin intermediate layer just below the meningeal layer of the dura mater. And then we have another layer which is adherent to the brain. It adheres to the brain. Okay. Now that layer is where that layer is what is going to be known as the pia mater. So the space between the pia mater and the arachnoid mater is what is going to be known as the arachnoid space, or the subarachnoid space is what is going to be known as the subarachnoid space. Okay, now in this subarachnoid space, that is where you're going to find the CSF flowing, cerebrospinal fluid. This is where cerebrospinal fluid does what? Flows. Cerebrospinal fluid leaves the ventricular system and enters the subarachnoid space between the arachnoid and pia mater. Okay, where it cushions and nourishes the brain. All right. So if you notice, this cerebrospinal fluid is leaving. Now, there are so many ways that cerebrospinal fluid leaves the ventricular system. So one of the ways is it goes through arachnoid granulations. There will be arachnoid granulations, these ones, okay, going into the going into the no sorry for the cerebrospinal fluid to move from the ventricles to the subarachnoid space, we are going to find that there will be places where this cerebrospinal fluid will be escaping. For example, in the fourth ventricles, we have some apertures that will allow for cerebrospinal fluids to move. So, for example, we have these apertures here, the lateral apertures on the sides of the fourth ventricle. And so this cerebrospinal fluid will move from there and come into the subarachnoid space and also go all the way in the subarachnoid space of the spine and circulates nicely. As it circulates in the arachnoid space, you're going to find that it is now going to start leaving the subarachnoid space and entering the the dural where there is blood. So as it is being produced, it will be produced at 400 to 500 mils a day, but it will still be coming out. It will come out into the subarachnoid space, circulate all through, and it will go back and it will get back into the venous system. So here in the choroid plexus, we have arteries that are bringing carbon dioxide, that are bringing oxygen and nutrients. And once the cerebrospinal fluid as it is produced, it contains the nutrients, it contains the oxygen, okay? And as the cerebrospinal fluid is moving around, the oxygen is being used up and it goes back into the bloodstream through the arachnoid granulations, okay? Into the sinuses, into the superior sagittal sinus over there. Okay, so that is how cerebrospinal fluid moves. So CSF leaves the lateral ventricles through the interventricular foramen. Remember this interventricular foramen, and enters the third ventricle, passes through the cerebral aqueduct into the fourth ventricle. Okay, some will leave this ventricle through the median and lateral apertures. On the lateral apertures, we have this one, and then we have this median aperture, okay, and enters the subarachnoid space, which is continuous around the spinal cord and posterior superiorly over the cerebellum. All right, posterior superiorly over the cerebellum. The main site of CSF absorption into the venous system is through the arachnoid granulations. We've talked about the arachnoid granulations. So if you look at them here, if you remove the skull on top, you've removed the peritoneal layer of the dura mater, and then you're just remaining with this space should be the sinus, okay? The durovenous sinus, and then there are these arachnoid granulations, and that is mainly how the fluid will be moving, the cerebrospinal fluid will be moving from the arachnoid space into this the durovenous space. Okay, so some of it will enter the venous system by transport through the cells of the arachnoid granulations. Some will move between these cells. Okay, that is how CSF uh, circulates. Then we have the functions of CSF. One is to protect the brain. This is to cushion the brain against blows of the head. Okay. Two is to provide buoyancy, and that prevents the weight of the brain from compressing the cranial nerve roots and blood vessels against the internal surface of the cranium. Okay. So because the brain is slightly heavier than CSF, the gyri on the basal surface of the brain will be in contact with the floor of the cranial cavity when someone is in an erect position. Okay, momentarily large changes in pressure occur during coughing and straining and during changes in position. Okay, and so that is what we're talking about. Now, the blood supply to the brain. So the blood supply is through two major routes. One, we have the internal carotid arteries, and we have through the vertebral arteries. Now, internal carotid arteries, they arise from the common carotid arteries, okay? From the common carotid arteries. We have external carotid and internal carotid arteries, okay? But we have this common carotid artery, which is coming from a branch that is coming from the aorta, okay? So once the common carotid artery starts ascending, there will be a point near the near C6 where the common carotid artery will bifurcate into an internal carotid and an external carotid. The internal carotid is the one that will go all the way up to the cranium, and it will enter the cranial cavity through a portion in the temporal bone, the petrous part of the temporal bone. Okay? Vertebral arteries, on the other hand, begin in the root of the neck as the first branches of the first part of the subclavian artery. Okay? As the first part of the as the first branches of the first part of the subclavian artery. This is the subclavian artery. This is the first part. We have the second part, and then we have the third part that side. So the first part of the subclavian artery, we are going to find that there is this branch on both sides of the neck. There is a subclavian artery on this other side as well. So you're going to find that there's going to be a branch which is going to be known as the vertebral artery. This will ascend up, okay, through the foramina that are in the vertebral bones, and they will go and pierce the dura mater and enter the cranial cavity through the foramen magnum. The foramen magnum is a very big hole, a very big opening in the occipital bone through which the spinal cord leaves the cranial cavity. Okay, so this is the foramen they will use to enter the cranial cavity. The intracranial parts of the vertebral arteries unite at the border of the pons to form the basilar artery. So if you look at this diagram here, we are going to notice that we have these two, we said on both sides. These are coming all the way from the subclavian arteries, and they're coming up, and then they're coming to unite over here. Okay, and then they're going, after they unite, they form the basilar artery. As they form the basilar artery, they give off branches. Okay, they give off branches that supply this portion of the brain. So this is what is going to be called the vertebrobasilar arterial circulation. Okay, as they go into on the inferior aspect of the brain, you notice that they are going to then divide. Okay, they are going to divide to form this posterior cerebral arteries. There will be a right posterior cerebral artery and a left posterior cerebral artery. Okay, now as they go, they are going to anastomose with a branch of the internal carotid. Remember, the internal carotid is coming from the body, and it is coming all the way here to join the circle of Willis over here. And then it will give off branches. Once these blood vessels, the posterior cerebral arteries, they communicate with the internal carotid, and then we are going to find that on the anterior aspect, as there will be a branch that will be given off from the internal carotid to form the anterior cerebral artery. Okay, and then a main branch will go from the anterior communicating artery. This internal communicating artery has been cut, because remember, it's coming all the way from the body. So this internal carotid artery has been cut, and so it gives us this. It continues as the middle cerebral artery, going to supply the hemispheres of the brain, the cerebrum, the cerebral hemispheres of the brain. On the anterior aspect, we're going to notice that there will be an anterior cerebral artery on the right and on the left, and these two communicate through the anterior communicating artery, a small communicating artery over there. So this is how, when you remove the brain tissue, this is the circular arteriosus. This is how the circular arteriosus is going to look We have the vertebral arteries forming the basilar arteries, giving off posterior cerebral arteries as well as posterior communicating arteries, which are going to anastomose with the internal carotid arteries coming from the body, from the all the way from the common carotid arteries, and then continue as middle cerebral arteries and give off branches of anterior cerebral arteries. The anterior cerebral arteries will then communicate with each other through the anterior communicating artery. So that is the blood supply of the brain. That is the blood supply of the brain. And so for today, we're going to end here. The next time we come, we're going to continue from where we have ended today.